Assalamualaikum, my name is Mimi Sofia and today I will be presenting about my proposal final year project entitled Effect of Extraction Solvent on the Antioxidant of Autocyphers Terminal Leaf Extract. And this is my thoughts layout.
First, introduction.
Autocyphers terminal is actually used traditionally to treat gout, arthritis, and inflammatory-related conditions. The in vitro anti-inflammatory effects of the plant have been scientifically investigated. That it is a Malaysian herb also known as locally as Nisan kucing and is widely grown in tropical regions. Staminus, or also we call as O. staminus, was extracted using a combination of traditional and modern method. This source is Asian region. O. staminus is actually leaves or harvested and dried to make a tea leaves.
Number 2, problem statement and solutions.
Staminus, a medicinal plant that has been used for millennia in traditional source is Asian medicine. It is to treat a range of maladies including urinary lithiasis, edema, eruptive fever, influenza, and etc. The goal of this study is to determine how the effect can be in extraction solvent on antioxidant activity of Autocyphers terminal leaf. So for the solution, many species in the genus have been remained underexplored or have been studied in a limited way, scientific validation with O. staminus being one of them. So the extraction of O. staminus on antioxidant activity were investigated, and also extract of O. staminus with various polarity were investigated. In the previous study, in vitro were also carried out to investigate the inhibitory action of the extraction compound in O. staminus leaves.
Number 3, objectives.
There are 3 objectives in this study. First, to evaluate the antioxidant activity of Autocyphers terminal leaves subjected to different extraction methods such as SLE, SFE. Second, to investigate effect of the extraction solvent on TPC, TFC, in vitro antioxidant activity of terminal leaves. And lastly, it is to synthesize Autocyphers terminal leaf using phytochemical tests and TPC.
Number 4, literature review.
For the chemical constituents, there are 2 active compounds that obtained in the O. terminal leaves. First, if you can see, the eupatorin compound. The compound eupatorin was able to reduce the number of viable cancer cells to the same extent as the extract with IC50 value in micromolar range. Second, you can see rosmarinic acid, which is we call as RA, it is the major compound of O. staminus, possess various pharmaceutical properties. In anti-diabetic, they have 2 studies. First, in vitro cell line study. For in vitro cell line study, according to the current in vitro data, animal infection study and scripton research, polymethylcellulose flavin from Java tea, which is O. staminus, have anti-diabetes action against eupogenic Escherichia coli, UPEC. Second, antioxidant property in vitro study. This is anti-diabetes action was reported to reduce bladder and kidney damage in a mouse infection model. In another experiment, investigated the antioxidative potency of various fractions of O. staminus extracts using an in vitro model of 1,1-diphenyl-2-picrihydrazyl salvaging. Of the results, observes that O. staminus show high antioxidant activity and could be considered as an immunomodulatory agent also.
Methodology.
For the methodology, first, the harvested seedlings were washed thoroughly under running tap water and separated into leaf, stems, and root. Second, the sample were dried in an oven for 3 days at 45 degrees Celsius. Next, all plant parts were separately ground into the fine powder and stored at room temperature at 25 degrees Celsius for further extraction. Fourth, the leaves were selected to determine the effect of extraction antioxidant activity. Next, leaves from the different treatment, 50%, 70%, and in open environment was harvested and washed. And lastly, the leaves were dried in an oven for 3 days at 45 degrees Celsius and ground into the fine powder and stored at room temperature, which is in 25 degrees Celsius for further extraction. So for the sample extraction, first 1 gram of each sample was extracted with 100 milliliter methanol at 40 degrees Celsius for 4 hours. Second, continuous stirring at 150 rpm using a shaking water bath. And third, the extract was filtered under the vacuum and cooled to the room temperature before freezing, which is approximately negative 20 degrees Celsius for the further analysis. And lastly, all the extraction were done in triplicates.
Number 7. Expected results.
Expected result of this study showed that leaves had the highest concentration of phenolate as well as antioxidant activity. All staminates grown under open environment exhibit higher TPC and antioxidant activity compared with the plant grown under 50 and 70% shading. Okay, so this is all of my reference. That's all from me. Thank you.